All right. Hello, everyone. Welcome to BOC Sciences webinar session. I'm Carrie Zhang, the Sales Director of BOC Sciences and also the host of today's session. I'm really glad you could join us today. For those who may not know, BOC Sciences is based in New York, and we are a comprehensive solution provider in pharmaceutical research and development industry. We specialize in everything from lipids and adjuvants to branched lipids and cholesterol. Our main focus today is on lipid nanoparticles for cancer medicine. In today's session, we are honored to have Professor Zheng present his groundbreaking work on theranostic lipid nanoparticles, specifically porphysomes and their applications in cancer medicine. Professor Zheng will explain how these liposome-like nanoparticles are making targeted delivery and multimodal imaging more effective, and update us on the clinical progress of LNP-based platforms in treating cancer. This is a great chance to gain some insights into the intersection of nanomedicine and oncology and discover new strategies for cancer diagnosis and treatment. As we embark on this exciting journey, I'd love for you all to get involved. Please keep in mind that during the presentation section, you will be able to post your questions related to the webinar content using the QA panel at the bottom of the screen. Now, settle into a comfortable and quiet spot to enjoy the upcoming presentation. Without further ado, let's give a warm welcome to Professor Zheng. You may begin whenever you're ready.
Thank you. May I share my slide?
All right. Please, I've already stopped sharing.
Okay. Can you see my slide?
Yes.
Okay, continue to share. Okay, thank you so much. Really appreciate this opportunity because I really love lipids. And what I like to share is our experience of developing a theranostic lipid, particularly a theranostic lipid nanoparticle, to really expand the scope of how that we can apply the lipid, not just for drug delivery, for gene therapy, but all the imaging and the entire scope of theranostics. So I'd like to start by talking about cancer nanomedicine. One of the hallmark of cancer nanomedicine is really the multifunction. Because many years ago, when the lipid nanoparticle started, the idea is really using the lipid as a way to formulate a drug and to improve the drug delivery, particularly to overcome the water solubility and reduce the system toxicity. Doxil probably the prime example of that, essentially doxorubicin loaded liposome. But later on, researchers have started to realize this lipid nanoparticle is an amazing platform to drive multifunction, particularly theranostic. For example, people has done by introducing multiple drugs, by introducing the contrast agent in the lipid nanoparticle to allow them for imaging guided therapy. And you can actually, using a different imaging mechanism, you can even incorporate its triggers for external release. So for that aspect, the lipid nanoparticle, multifunctional nanoparticle, not just lipid nanoparticle, but a multifunctional nanoparticle per se, has been play a huge role in the cancer theranostic, in imaging guide drug delivery. You can actually for track that patient for particular cancer therapy, so on and so on. But multifunctional cancer nanomedicine usually is actually most focused on, I would say 99% of them are focused on this all-in-one concept, essentially taking different functional building block, put them together like a Lego that allow you to do different functions. So this is all-in-one concept. This is very advantageous by incorporating the clinical approved component together. So reduce the clinical translation hurdle. But on the other side, because of the multi-component, it really induced the complexity and actually introducing an uncertain level of in vivo properties and the reproducibility, toxicity, and the rest of the concerns. So we were actually thinking, many other groups as well, about 15, 20 years ago, we are starting thinking, how can we change that approach? What if we're using a multifunctional building block to build this nanoparticle? And that the functional building block itself is multifunction. When they put them together in the nanoparticle form, you give them additional nanoscale property. That will be very nice because it will be much simpler for the translation, so we thought. But the challenge of that is there's not many multifunctional building block existed so at that time. So this is the reason why the cancer nanomedicine so far, most of the approved drug is still liposome-based, lipid nanoparticle-based, because of as I mentioned, the multifunctional is hard to achieve with complexity. That is the reason why the lipid nanoparticle mostly still using for drug delivery, right? Nowadays, LNP lipid nanoparticle has been getting so much strain in the gene therapy, and particularly with Onpattro, the approval in the first siRNA lipid nanoparticle, followed by the major advance in the COVID vaccines that lead to the everybody's exploration into LNP space. And actually, indeed, even the CRISPR editing has shown very promising results in cancer nanomedicine. But these LNP classically are made by the 4 different kinds of lipid. You have a PEG lipid, HELP lipid, ionizable lipid, cholesterol, plus a complex with a nucleic acid. So in terms of the lipid nanoparticle for multifunctionality, I think this is one of the best examples I want to give is from my colleague, Christine Allen of UT. Basically, a liposome and classic all-in-one concept. You put in the drug inside the middle. You can have a contrast agent, CT contrast. You can have the nuclear radioisotope imaging, optical imaging, and targeting a classic lipid nanoparticle in all-in-one concept. The question is, can we make a multifunctional building block, lipid building for all 1,4 nanoparticles? That led to us into about 15 years ago, we started this venture. We're interested in the porphyrin because porphyrin are considered a color of life. It is actually existing in your body, make your blood red because that's hemoglobin. Hemoglobin, the component of hemoglobin, the heme. Heme is iron porphyrin molecule to make the blood red. But mostly importantly, transfer oxygen make human being human. You also see that all the grass is green, the tree is green. Why? Because of the chlorophyll. Chlorophyll is a magnesium porphyrin. So you won't, I think I make a strong argument porphyrin is the color of life. But more importantly, porphyrin in the biomedical application is well known as a fluorescence imaging agent because the porphyrin is fluorescent. It is probably most well known as a photodynamic therapy agent because porphyrin interacts with the light and oxygen, generate reactive oxygen species that can lead to the killing of different target cells. Less well known is porphyrin's metal chelation property in nature can be hijacked for imaging purposes. For example, if we're replacing the magnesium in the chlorophyll or iron in the hemoglobin, we use that for radioisotope. It gives us the PET imaging. Basically, you can transform porphyrin from a purely a biphotonic material into nuclear masking space. We can also using porphyrin to chelating aromatic metal, for example, manganese, that allow for the MI contrast. So I hope I build a case. Porphyrin is a theranostic building block. Now let's lead to the central of my talk today. It is we discovered about 14 years ago, porphyrin, when the porphyrin replacing one of the fatty acid chain of the lipid, phospholipid, basically forming this porphyrin lipid molecule, essentially synthesized from lysophosphate conjugation with a porphyrin molecule, it gives this porphyrin lipid molecule. We find out this porphyrin lipid molecule can self-assemble just like the lipid self-assemble liposome, porphyrin lipid self-assemble into liposome-like porphyrin lipid nanoparticle we call porphysomes. Initially, this is actually, we never expect that happen. It could purely a serendipitous discovery because porphyrin is a bulky molecule. We never thought the porphyrin lipid can self-assemble. But if you look at the diagram below, you will see porphyrin, the hydrophobic nature of the porphyrin, make the porphyrin less soluble, become a stabilizing force. When it's pi-pi interaction, make the porphyrin become a stabilizing in the liposome bilayer. We call this porphyrin bilayer. That leads to an entire journey down the next entire talk today. So the first thing we think about is when you have 180,000 of these porphyrin lipids self-assembled in a liposome-like structure, what happens is this porphyrin, every porphyrin absorbs light. When you have 80,000 of them, they absorb tons of light and they quenched, just like a black hole does. What a black hole does is the light sucking, nothing come out, only came out by heat. This porphysome behaves the same way. All the light comes in, it becomes extremely photothermal capable. So we can use that for the photothermal ablation therapy and actually beats the inorganic nanoparticle, the gold, the carbon nanotubes, we can actually achieve as efficient, or if not more efficient, photothermal therapy efficacy, as you can see on the example on the right. Another interesting thermal conversion property is called photoacoustic. Photoacoustic means, photo means light, acoustic means ultrasound, a wave. So what happened is, instead of using a continuous wavelength laser that will absorb, keep absorbing the light, that's powerful, then the temperature keep rising. Now you're using a pulse laser. Pulse laser means it didn't only generate transit temperature increase. The transit temperature increase lead to the thermal expansion, lead to the bunch of wave coming out that be detected by ultrasound. So the photoacoustic is also a thermal conversion. The difference here is light in, sound out, so you can have very deep tissue penetration. Like for example on the left, we show the metastatic lymph node can be entire lymph node can show homogeneous distribution. Basically, we see the light detection of the biphotoacoustic. So high resolution imaging. And as I mentioned, porphysome is not like a rock, it's not like a nanoparticle. It behaves like, I would say, liposome, essentially. And liposome, we know, is a dynamic. There is always dissociation of lipid nanoparticle from some monomer the properties. And the porphysome is also dynamic. So if you have a small portion of the porphysome can be dissociated in the tumor, the porphyrin monomer property can be restored, so we can achieve the photodynamic therapy efficacy as good as FDA-approved photofree. We can also using the fluorescence capacity of the porphyrin monomer, and that lead to the fluorescence guide resection. In the example on the left, we show in the endometrial cancer case, we can using the fluorescence guide resection and to actually remove the residual tumors, we actually achieve the detection sensitivity of 98.4% and the specificity of 80%. Another interesting phenomena, as I mentioned, is the porphyrin monomer can be hijacked for the PET imaging. We demonstrate the preformed porphysome nanoparticle can be used directly to radio label the copper-64. Bear in mind, the copper-64, the radioisotope, is not chelating, it's not captured in the nanoparticle. It is captured in the building block, the porphyrin building block of the nanoparticle, which means what you see is what you get. Not only you can track the nanoparticle movement, you can track when it's a nanoparticle dissociate, become a monomer. For example on the right, you can see we can detect the nanoparticle. We can see the porphysomes, copper-64 porphysome light up the oral tumor in the rabbit like a live fireball. But what's amazing is this fireball, you can see the draining down the lymph node, which we confirm is metastatic. So that means not only we can using the fluorescence, porphysome fluorescence to actually tracking the tumor to the lymph node, we can also see in the primary tumor to the metastatic lymph node by the PET scan. As I mentioned, porphyrin is known for MRI as well, because when you're chelating parallel metal, but now you're taking advantage of the porphysome. Because when you talk about 80,000 porphyrin molecule, imagine you have 80,000 manganese porphyrin. That means you have overcome the sensitivity limitation of MI, which is one of the key hurdle in the MI detection because of poor sensitivity. And this way, we can actually use that not only as a light absorber, because it still absorbs light, you can use in the multi-modal imaging. So in essence, this diagram demonstrates porphysome is made by a single porphyrin phospholipid molecule, about 1,000 molecule weight, self-assemble into liposome-like structure, and it can achieve all the function at the same time, or simultaneously or subsequently, which means porphysome is really the first-in-class theranostic lipid nanoparticles. And with this in mind, we actually take advantage of the intrinsic multifunction, build a very unique phase 1A, phase 1B clinical trial protocol. The idea now is because it's the first-in-class molecule, we want to make sure it is safe to use in patient. So when the first in patient, we actually have the maximum precaution. So the idea is to start with the PET imaging with the copper-64 porphysome for the advanced ovarian cancer patient. And this imaging study will allow us to show the safety, to actually demonstrate the pharmacokinetics, to demonstrate the tumor targeting. Once that is all done, we can move on to start the much expandable umbrellas theranostic safety study, try the different function of the porphysome in the different cancer type simultaneously. So that is idea. So why we start this porphysome? Why we start this porphysome for copper-64 porphysome first in human? Because the copper-64 porphysome is a tracer, the copper-64 is a tracer. That means it labeled such a small amount of the porphysome ring, porphysome structures, it allows us does not alter the parental porphysome property. We actually demonstrate, not only assume that, we actually validate that by compare with a copper-64 porphysome versus a porphysome in terms of structure, function, the in vivo behavior, the pharmacokinetics, biodistribution. Basically, we demonstrate the copper-64 porphysome and the parental porphysome are physicochemically, pharmacologically, and toxicologically equivalent. This allows us to demonstrate. This also allows for the safety profile of porphysome in rats and dogs. We did not raise any concern in terms of their use in human. We even did the monkey study to demonstrate on the biocompatibilities. This radio-labeled copper-64 not only allows us to accurate imaging-based assessment of the PK, is really using that very useful predict the porphysome using for other non-radio isotope-based indications. So another reason why we want to start the porphysome, copper-64 porphysome first in human is really exactly because of pharmacokinetics. Using the machine learning approach, we actually demonstrate copper-64 porphysome PET imaging can serve as a dosimetry tool for the parental porphysome. This is a study we done in the mice. We basically demonstrate if you take a PET scan for only a few organs using the machine learning we call nanomask protocol, which is open access, we allow them not only to imaging analysis for auto segmentation, but more importantly, it predicts the pharmacokinetics in the multiple time points and in the multiple species. So this will become very handy when we actually design them for the prediction of the nanomedicine pharmacokinetics using a limited PET scan in patient. Why we believe in the advanced ovarian cancer patient, the data can be applicable to other cancer types, because we also demonstrate across different cancer type, different animal species, and the different models, we demonstrate the copper-64 porphysome PET reflects the ubiquitous thermal selectivity of the parent porphysome in the many different cancer types. So for example, here in the top row, we demonstrate the mouse and the rat and the rabbit and the canine in the prostate cancer and oral cancer. We demonstrate the metastasis and everything demonstrate in the selectivity that way. So we also encountered a lot of translation hurdles, and one of them particularly a lipid chemistry based. And I want to give you this example. During the time we synthesized the porphyrin-lipid and called pyrolipid, and we're actually using that for assembly of the porphysome, we observed because of the phospholipid has radioisomers, SN1, SN2 isomer, which correspond in different phospholipase activity, but we call SN1, SN2. But when the phospholipid fatty acid chain is become the porphyrin, it becomes very bulky, as you demonstrate on the left. This bulkiness of the porphyrin ring make the isomers become separable even by HPLC. But once we separate this radioisomers, we observe they have entirely different in vivo fate. Essentially, for example, on the figures here, you can demonstrate in the SN2 isomers, while they are stable in the serum, in the blood circulation, allow the nanoparticle to the tumor. Their spleen and their liver, within 12 to 24 hours, majority of them are starting to be cleaved by phospholipase A2. But in the same one isomer, they remain non-metabolite for at least 40 hours later. So take a week for them to degrade. Initially, we saw this is a fascinating result. We actually published in the paper, but later on, we realized this become a nightmare in the translation because Health Canada and the regulatory agency is going to ask, you cannot have two isomers and keep changing the ratio in order for translation. So the answer, the question we actually have in the demand we have in hand is either we make an isomers with fixed ratio from a different scale, a milligram to the gram to kilogram, or we find one isomer, get 100% this isomer and stick with it. And take us 3 years to figure out the synthesis, actually to make out this SN2 isomeric pure, about 99% of this isomer pure at 200 gram synthesis. So this is really another lesson we learned, is the translation is much, much harder for the field. So where are we Porphysome now? We achieved the GMP Porphysome synthesis at the BC Cancer. Initially, we actually interested in getting the CRO in the US to do this, but during the COVID time, everybody is shifting to the COVID vaccine. So we only can work with academic partners at the BC Cancer, and we got this GMP Porphysome made. And for the copper 64 label Porphysome, we actually did in our pharmacy department across the street. So it will be only 200 meters from where the Porphysome will be labeled, then to the patient injection, which will happen this month. So now I want to move on to talk about, this is really the concept Porphysome really start the floodgate of theranostic lipid nanoparticles for cancer medicine, because Porphysome is serendipitous discovered, right? However, we want to expand this knowledge of the porphyrin lipid can self-assemble, expand that to a family of the lipid nanoparticles families. For example, we demonstrate we can using the HDL mimicking Porphysome, which is called porphyrin HDL, having the size of only 10 nanometer and without pagylation, it actually can be mimicking, serving as a true surrogate of a native HDL. We demonstrate that utility, imaging and therapeutic utility in the cardiovascular space. We demonstrate, for example, porphyrin lipid can be using to self-assemble into porphyrin microbubbles that allows the ultrasound-induced microbubble field to actually expand to multi-modality and give us the way to monitor how the bubble becomes the nanoparticles and how the bubble can be used for gene delivery of different assays. We even made a proto cells, essentially an artificial cells made with porphyrin lipid called giant porphyrin vesicles, allowed to do the molecular reaction inside this aqueous core and outside in the layer of the surface. But I want to give you a few examples today to really to show you how you change a different lipid with a dye can actually make a different functions. The first example I want to give you is called J-aggregate Porphysome, J-Porphysome. This idea is coming from the nature of the photosynthesis. And this is the photo I took from Yellowstone when I visited there. and it was beautiful geyser and a beautiful color. And the color of this Yellowstone hot spring geyser is actually most interesting because they are most enriched microbial environment in the world. There's many discovery made in this one, for example, the PCR enzyme. But what I'm interested is this photosynthetic bacteria called blue-green algae called cyanobacteria. If you look at the cyanobacteria, they are these, if you look deeper, you will see they are each individual of them is called photosynthetic membrane, and go deep enough, you will see arranged by this called, in the middle, it's called light harvesting complex. And this light harvesting complex is demonstrate a fascinating behavior. That is a single porphyrin, in this case called bacteriophyll molecule, the red and green are chemically identical. But when they are spatially arranged in the way are different by the protein matrix of the photosynthetic membrane, they change the entire optical spectrum. For example, in this picket fence form, these bacteriophyll, they have 850 nanometers. But when they are in the spatially distributed form, they have 800 nanometers. So this basically gives you a single molecule, a porphyrin, give you entirely different wavelengths. Nature is fascinating to design that. It's almost like one nothing computer binary code. So we were actually thinking the bacteriophyll had this big fatty chain next to this porphyrin. In the protein matrix, they hold them in the different spatial orientation. So our question is, can we capture the magic of bacteriophyll in the photosynthesis in a lipid nanoparticle form? For this, we made this bacteriophyll phospholipid, similar to the porphyrin-lipid, phospholipid in the porphysome case. But here, we try to mimicking the protein matrix instead of entire self-assemble of the bacterial lipid. We put this bacterial lipid basic insert back to the lipid matrix, like for example, 15%. So the J porphysome is made with 15% bacterial lipid in the regular phospholipid. Here, because of the matrix allow the bacterial lipid to self-assemble in the ordered aggregate. For example, here, we can observe this bacterial lipid form a beautiful J aggregate in the membrane that lead to a 75 nanometer redshift from the A750 nanometer all the way to A25 nanometers. And you can see the secondary structure showing the CD spectrum and showing the fluorescence of the classical J aggregate, and very nicely characterized. And using this J aggregate, we allow us to show a much deeper red shift. So give us a beautiful dual photoacoustic imaging of the sentinel lymph node mapping. For example, injection here, we can light up a sentinel lymph node. We can see a beautiful photoacoustic imaging corresponding to the fluorescence imaging. But more importantly, we discover the stimuli responsive behavior. Imagine we have a bacteriophyll, the aggregate inside the J porphysome. But when this is inside the lipid matrix. Lipid has transition temperature. So when you heat up this transition temperature to the transition temperature, the lipid membrane becomes more fluidity. The fluidized of this membrane allows the J aggregate becomes monomerized, becomes disaggregate, and with maintaining the nanoparticle structure. So you are still liposome structure, but one with aggregate J, one is a dissociated J. And this can be, transition temperature can be tuned by using different phospholipid. That's actually fascinating because that's one is tunable with the temperature. Two, it is actually showing a beautiful the temperature response. As soon as you're hitting the right temperature, essentially, to make them disaggregate, you will see the instant drop in the photoacoustic signals, very rapid drop that allow you to do the sensing purpose. Number two, this is actually reversible. This, in fact, is the very first reversible photoacoustic sensor. What you see here is because it's a temperature response, in the cold state is 824 nanometers. We put in the PCR machine during the cycle, we can see when the temperature increase, if the wavelength shifts to 750, when the temperature cool down, it go back to 825. You can keep doing the cycles. So tunable, reversible, and now we can actually get into why we need that. It allows us to do a thermal sensing, for example. Here, I have a two dye in a tumor in the mice. The red one is indocyanine green, which is not temperature responsive. The green one is our J porphysome. What I want to show you here is by a set temperature, if we hit them with a light, then temperature goes up. As soon as you're hitting the set temperature, in this case 43 degrees, you will see the green disappear. Bear with me. The green disappear. This is literally means we have a temperature sensors. When you actually in the Thanksgiving, you cook the pop timer people using, essentially what the mom will tell you, I don't need to know the temperature, I just need to know pop up, the temperature's up, surgeon will like that. But this photoacoustic sensing mechanism can be extended to different biological stimuli like pH, enzyme, and so on. Another interesting phenomenon we actually find this one is with this sensing mechanism is actually a built-in thermostat like air conditioning. If I set temperature, the temperature will not go further. Because if you look at the top line, whenever a laser absorbing by the dye, right, they will generate heat. But the problem is the heat didn't go up, didn't stop. So overheating, the light going deeper, that's a light attenuation. So this way have a major issues. But when you're building a thermostat, we know the temperature goes up. As soon as you're hitting the set temperature, it disaggregates. That means the entire path cleared out for you. So you actually can indeed, almost using this analogy, you actually, like people are crossing the wall. As you crossing the wall start open, like Harry Potter. Once you're passing, the wall closes behind you. So this is the smart photothermal therapy ideas. So now I want to shift gear to another example. We want to expand the nuclear medicine space in terms of metal ion chelation into the theranostic space, into radiotheranostics. We know the porphyrin has difficulty to chelating anything beyond the diode cut time because the pocket of the porphyrin is not big enough to the 3+I. But there is a many idea. People are trying to make the porphyrin with chelating different metal like lanthanide. This is one of the best examples. Essentially, you can using a stabilizing ligand to make the lanthanide stabilized by the porphyrin, which is complicated. But eventually, people are using this methodology for chelating European terbium for these two-photon imaging. But I want interested in the simple idea. For example, Professor Jonathan Sessler at University of Texas came up with this brilliant idea. He synthesized an expanded porphyrin called taxophrin because he's in Texas, so he called taxophrin. And it's also because of the 5 coordination from a star called Lone Star State reflecting Texas. And this 5-star coordination is important because not only you have the 4 become a 5 coordination, you also gain a 20% larger pocket. So you can chelating majority of transition metal, particularly lanthanide. So we take a while ago, figure out a way to synthesize another lipid. This time we make a taxophrin lipid molecule. This molecule, in fact, is hard to make. That's why we got all the patent issued. We spend a lot of time trying to make this one work. But once this taxophrin lipid molecule is synthesized, the floodgates open. We can make the self-assemble just like paralipid did, form this called nanotechnology. Now you can stable chelation of entire spectrum of lanthanide in plus a transition metal. For example, Samonian, European, Galenian, terbium, holmium, many different things. And this is important because the radiotheranostics has become a huge hit in the medicine, cancer research, not in the cancer patient care, for example. And we demonstrate for radiotherapy, we can actually incorporate alpha emitter of lead-201, 212, bismuth-213. The beta emitter is very workable, feasible using the yttrium-90, lutetium-177, like the drug in Lutathera, and terbium-161, and OJ electron emitter, which is indium-111 and thallium-201. Essentially, we can apply this taxophrin nanotaxophene, using this taxophene lipid for multiple cancer applications. Let's just give an example. And to make this more easy, we actually show these can be custom metal chelated using different labels. We even develop our own disposable kit for the radio labeling of the taxophene, nanotaxophene, and also for radio labeling, show you this disposable kit to generating a nanoparticle on that. Essentially, you can buy the chip and we can creating the syringe pump. It allows us to instantly making the radio label hot mixing, as well as to do making the nanotaxophene nanoparticles. We demonstrate using 177 lutetium for the SPECT imaging, indium 111 for taxophene for AJ. We also made the lutetium taxophene for photodynamic therapy using the cold lutetium, and gadolinium taxophene for MRI. And another example we're using is for the radiotherapy, radiosensitization, because we know when Jonathan Sessler in Texas synthesized the taxophene, he actually made this taxophene all the way to phase 3 clinical trial as a radiosensitizer. Unfortunately, it was not approved by FDA after 1,000 patients because of the not enough radiotherapy efficacy. But the message for us is gadolinium taxophene is safe for a synthetic molecule and is only has a modest radiation sensitizer efficacy. So how can we recapture that to actually make it work? My student, Professor Zheng in the Peking University, and together with my collaborator, came up with the idea. They make this taxophene lipid to self-assemble into nanotaxophene, incorporating the microbin, the oxygen carrier. So give you the two ideas. One is using the maximum loading of this radiosensitizer, we want to improve the radiotherapy efficacy. And the other idea is to carry the oxygen to improve the synergize with the radiation therapy by relieving the hypoxia. And this method has been worked very well in terms of relieving the hypoxia by absorbing the oxygen in the primary capillary delivered to the tumors. And we demonstrate through the collagenic assay, we demonstrate significant radiosensitization efficacy compared to some of the standard radiosensitizers. And this method has been pretty powerful. So now I want to shift gear to the another example. This is actually involved another interesting lipid is called EDTA lipid. So I mentioned porphysome is self-assembled from a porphyrin lipid. While the porphyrin lipid is very good for photodynamic therapy, porphysome is very good for photothermal therapy, and there is a dynamic process, so there's some parties associated. But porphysome itself is already effective PDT agent, even a very small portion exists in the dissociation form. But the biggest problem of this porphysome for photodynamic therapy is it has very low intracellular uptake, just like a liposome. Liposome doesn't get take up cell very well. They actually majority of them still in the tumor in microenvironment. So want to see how can we find a way to a new strategy to improve this therapeutic intracellular uptake, a general strategy, for example. So we're actually looking at this called EDTA, which is a very used molecule in the chemistry 101 for the titration, for the experiment. We're also using for clinically approved for treating the heavy metal poison because it can chelating the metal very efficiently. Another interesting phenomenon of EDTA is been using to increase the permeability of the gram-negative bacteria by chelating the dialcation, for example, magnesium, calcium from the bacterial cell wall, make the bacterial cell wall more easy to penetrating by the antibiotics. So we thought, this is actually maybe an idea. Can we, but this one is in the millimolar concentration. So the very, very ineffective. Obviously, you're not using millimolar concentration of EDTA for a nanomedicine approach. So we were thinking, can we make an EDTA conjugate to a phospholipid? And once, not phospholipid, conjugate to a lipid. And this EDTA will be part of the composition of the porphysome, essentially homogeneous distributing between the porphyrin lipid. This will allow the antimicrobial property to be improved. And we demonstrate with this EDTA lipid, we demonstrate a very, very significant antimicrobial activity, can kill the bacteria very efficiently, and with only 50 micromolar EDTA lipid, which compared to 5 millimolar is reduced the concentration by 100-fold. Then we take this idea further. We want to see, can we move this for cancer, improve the cancer cell uptake? So for this, we did a lot of optimization for the ratio, different percentage of this EDTA lipid and its porphyrin lipid. We demonstrate across the board, we can see significant improvement in the intracellular uptake. That's very interesting. Then we actually want to see if it is the mechanism of action is still the chelation of divalent cation. The answer is absolutely no. Because, for example, we know the EDTA lipid, EDTA chelating calcium and magnesium, but DTPA does not, because it's DTPA chelating gadolinium for MI contrast, for example. So if the EDTA and the DPA behave exactly the same way, so that means it may not be because of the metal chelation property. That's evidence one. Evidence two, when we insert the gadolinium into this metal, into the DTPA, and this does not change the uptake property at all. So that means metal chelation is not a property. Third, we actually do the excess competition with the calcium, magnesium in the EDTA lipid. We actually show no change at all. So that means it is not because of the chelation of dimetal. It is involves some very unique novel mechanism. And we're actually able to identify this mechanism is called membrane fluidization. Essentially, what does is EDTA lipid behave like a detergent-like property to make the membrane of the cancer cell become more fluidized that allow the nanoparticle or like porphysome to get inside the tumor cells. And to use this methodology, we demonstrate a very, very significant improvement in the fast activation of this EDTA porphysome uptake in cancer cells. Compared to the porphysome, we can achieve 15 minutes, we can see the tumor cell uptake. This is not the subcutaneous tumor xenograft. This is a spontaneous tumor, carcinogenesis induced in a hamster cheek model. We can show this fast activation. We can demonstrate in the tumor, see the much improved photodynamic therapy effect. And this is, more importantly, we actually demonstrate later on to show this EDTA lipid enhanced intracellular uptake is generalized in other lipid nanoparticle, for example, doxorubicin, doxo. So we can improve using EDTA lipid to improve the efficacy of doxo using this unique mechanism of intracellular uptake enhancement. I want to use the final two slides to wrap up my talk, because porphysome was made with different porphyrin lipid. We can expand the porphyrin lipid to different porphyrin lipid, for example, the bacterial lipid, the taxophrin lipid, or the different kind of lipid. But we also want to show porphysome, can we make a porphysome without porphyrin lipid? And this is the idea we actually made. We synthesize this azi or DP lipid. We find out they self-assemble into the liposome structure, just like a porphysome. We name bodipsome. That one interesting phenomenon of this bodipsome is, first of all, they're not porphyrin, so we now cannot call it porphysome anymore. But most interestingly, we find out that instead of J aggregation from the entire in the secondary high order secondary structure, they only form dimers. This bodipsome only form dimers, reflected in there's no peak shift in the J aggregation, even though we have a secondary structures. And this allows us to show very high photostability. Because of this property of the dimer, their unit is single dimer, not a high order structure. And that allows us to show very high optical stability for simultaneously photoacoustic and fluorescence imaging of cancers. So that's 45 minutes, and that will be my summary. Porphysome, so far I have shown Porphysome, an intrinsic theranostic lipid nanoparticle, has all the function detailed below: the photothermal, photoacoustic, photodynamic, fluorescence when disrupted. The metal chelation really transformed them for the biphotonic nanomaterial into MI, PET, SPECT, and radionuclide therapeutic agent. Porphysome tumor selectivity is preserved across cancer type, and now we're entering the first in human. And the next generation porphysome, when we expand the porphyrin-lipid into different porphyrin-lipid, or expand the porphyrin-lipid, or even non-porphyrin-lipid, it can expand the great scope of the theranostic lipid nanoparticles. So that's actually my talk, and I'd like to thank my lab members, particularly my first group of the graduate students from Toronto about 10, 15 years ago. These are, most of them are faculty member in academic now, and they call themselves the gangsters, right? So they're taking my name, and I'm very proud of them. And also thanks for all the funding support, particularly the program able to pushing all the way from discovery to the clinical translation to first in human is because of the tireless effort, especially by our Princess Margaret Cancer Foundation and Terry Fox Foundation for the support as well as the grant. And thank you very much for your listening, for your attention, and happy to answer any questions.
All right, thank you very much, Professor Zheng. That was an enlightening presentation. I believe all of our attendees found it as fascinating as I did. Now let's move on to the Q&A session. Professor Zheng will address some of the questions in the Q&A panel, and to all of the attendees, you're free to post your questions in the Q&A panel at the bottom of the screen. I do have a question about your work. I mean, have you done any studies on the regulation of drug release rates from the porphysomes? Like how can the formulation of the porphysomes to be adjusted to modify the drug release rate?
It's a very good question. Two answers. One, my former PhD student, Professor John Laval at the University of Buffalo, has actually so first of all, porphysome, when you have a lot of porphyrin-lipid inside the biolayer, they make the biolayer more rigid. So some of the drug inevitably will be released, will be actually leaked out. So in terms of the encapsulating of the drug, small molecule drugs, porphysome is not ideal because of the leakage of the rigid membrane. However, this can be overcome by decrease the percentage of the porphyrin-lipid in the liposome structures. When using 1 or 2% porphyrin-lipid, John Laval demonstrated you can use that for very efficient light-triggered drug release for the doxil for many other drugs. So that's one angle. The second angle is we are using the introducing the porphyrin-lipid as a trigger into the lipid nanoparticle for siRNA delivery. And we demonstrate by using this laser-activated drug release, we can actually improve dramatically the cytosolic release of the RNA into the cytosol, improve efficacy. So these can be very useful if potentially, for example, if we actually develop mRNA vaccine using the local delivery of this mRNA vaccine, and you follow by the light trigger, you could imagine having a on-demand trigger to release mRNA into their disease site, right? So I think there is a very useful for using this, but these are the very understudied, I would say.
Okay, thank you. And another question is about like, have you done any research like the remodeling of the extracellular matrix in certain tumors? Will they also affect the permeability of our porphysomes? Have you done any research on that?
Yes, yes or no. We were actually active in discussion to move into the extracellular space. But one thing we know is porphysome is in the thermodynamically favored state. what I mean that is, if I have a porphyrin microbubble, if I release the gas from the bubble, it actually becomes porphysome. When I have a porphyrin HDL, if I'm using the protein escape to digest the protein to actually bind down this nanoparticle, a small particle become a porphysome. There is a very little exchange between the serum protein and the porphysome because of this, exactly because of the porphyrin interaction in the bilayer hold the porphysome in a thermodynamic favorite state. But does not necessarily mean porphysome is like rock. They still dissociate, right? They're always a small portion they dissociate. But this dissociation can be taken advantage or trigger as activation mechanism as well. Yes.
Okay, thank you very much. Well, I have another question that might be not that related to Porphysomes. I wonder, could you please share your insights about the development trends in the formulation of nanoparticle lipid drugs, like the formulation of LNPs? Could you please share your insights about the development trends of this formulation?
Yeah, so it's an evolving process. So I will say share one examples, a different kind of like, so when we initially designed Porphysome, our idea is we want to take advantage of all the knowledge the field has already developed. We don't want to reinvent the wheel. So we follow exactly the Doxil formulation, like 5% PEG lipid and other thing. If I do it again, I would probably not do that. I will stay away from the PEG lipid because the PEG lipid may actually have unintended consequence in some circumstance, right? So we were essentially give us many extra effort to prove the toxicity in the monkey, in the dog, in all sorts of things, because there was some infusion reaction we encountered based on the PEG lipid. And in terms of how we formulate, but in fact, the HDL porphysome, the porphyrin HDL does not contain the PEG lipid. So, if I have a chance to redo all over again, we probably will choose a different porphyrin lipid nanoparticle go forward. That's one. Second, in terms of how we formulated, that's exactly what we actually did. Initially, we followed essentially the size extrusion. We even actually bought the big size extruder, high pressure extruder. But now we actually switching to industry standard as ethanol injection method to make the lipid nanoparticle that way. And this is our now preferred mechanism to go for the GMP production.
Okay, I see. That's really valuable sharing. Thank you very much. And let's see. I think we have some questions from the QA panel. Maybe Professor Zheng, you can answer them. You can choose some questions, you can answer them.
Yeah, I think the first question is giving the dual imaging capacity. What's the biggest challenge in scale, their translation, particularly regarding regulatory approval and the larger scale manufacturing? For larger scale manufacturing, we actually spend a lot of years effort. We were now able to making 200 gram synthesis batch for the porphyrin lipid building block. That allow us to actually move to, in fact, a few synthesis, we can get a 500 gram, half kilo of the porphyrin lipid, and they are single isomers. They allow us to do translation scale up is actually a problem is actually resolved because the porphysome, as I mentioned, the actual formulation follow the classic lipid nanoparticle manufacturing. So we don't want to reinvent the wheel. So the large scale manufacturing, the bottleneck is a monomer building block synthesis, which takes about 3 years to solve that. In terms of regulatory approval, this is a brilliant question. That is, initially, we thought that intrinsic multifunctional nature of the porphysome will give us a leg up into pushing the translation into different space, but we were wrong because as a first-in-class material, you're facing as stringent as a new drug in the FDA, in the Health Canada eyes. I think the translation expectation will happen after our first in human, after we fully demonstrated safety. Then, because of the intrinsic multifunction, we can take advantage of that for all different cancer types. All that we already done a lot of groundwork with that. I think that's probably my answer. The mechanism. Okay, so the second question, evaluate the mechanism of why nanoparticle accumulate in the cancer site. This is a brilliant question. I got that every single talk, every single time. We used to having trouble to answer that question because we always thought it's because of we feel inadequate to explain this by demonstrate because of nanoparticle EPI effect, enhanced permeability, because of passive accumulation of tumor. We feel inadequate to use this mechanism to explain why porphysome was so selective, going to so many different cancer types, different species. And later on, we figured out, we have a hypothesis. Our hypothesis is really based on the lipid, based on the radioisomer of the lipid. So the idea is the radioisomers, we have a different SN1, SN2 isomer, they cleave by different lipid, different phospholipase. And because we happen to have a single radioisomer responsible for phospholipase A2 to be cleaved, specific cleavage, that gave us an enzyme activation mechanism to go with the passive accumulation. So my belief, it is a combination of passive accumulation of nanoparticles in the tumor environment together with the enzyme activation, because phospholipase A2, particularly the secretive one, is actually expressed in tumor microenvironment. And the porphyrin, once it's cleaved, they have natural ability to get into the cancer cells. The third question is, let me see. Oh, thank you so much for your kind word. Are there any, so final question, are there any other application for these nanoparticles? Yes, the answer is unlimited. We're still in the exploration stage. For example, we demonstrate these porphysome in combination with a light activation maybe at different light dose than what we do PDT, photodynamic therapy, it is generating very robust immune response. So that is very exciting for us because now we can imagine we can actually drive as a light therapeutics. The bigger limitation is using the local cancer control. But once the immune system involved, we may actually have a very high possibility to expand that into the disease, but disease control beyond local. And also we demonstrate we can now introduce in different stimuli, for example, the X-ray, for using porphysome improve the radiation sensitization. So there's many different ways we actually are starting exploring. And the story of the porphysome has not ended, I would say. I think there is more to explore. Thank you. I think that's all the question I got.
All right, thank you. Thank you. This wraps up today's webinar session then. And we sincerely thank you for your time and attendance. And a special thanks goes out to Professor Zheng for sharing his valuable insights with us today. And BOC Sciences, we regularly invite experts from various fields to share their latest findings. So please keep an eye on our official website for upcoming webinars. And also, I want to remind you that our team is here to assist you with any inquiries you may have regarding our brands, our products, and the future webinar sessions. And we encourage you to connect with us and explore partnership opportunities. Thank you once again for being with us today. Stay safe, stay healthy. We look forward to seeing you also in future sessions. Thank you once again, Professor Zheng.
Thank you. Thank you.
Okay, that's it then. Bye.
Okay, bye-bye.